looking at the. Okay, good morning to everybody. We're here with Dr. Jose Manuel Santa Cruz, and we want to present, and we have the honor.
Y tenemos el honor de presentarles al doctor Kosik de la Universidad de California.
And he is visiting us regarding the Red Lab project, and of course some projects in the future that we will have with him. If Jose Manuel wants to give him the welcome, has to be in English because we are in the English channel.
Hello, welcome all. We are here with Dr. Kosik, and we are in Intelectus, Centro de Memoria y Cognición in Bogotá, Colombia. Welcome all.
Okay, so we thank Dr. Kosik and we give him the word to him.
Good. Okay. Thank you, everyone. Hola a todos. I'm very happy to be here. My name is Ken Kosik, professor of neuroscience. And I'd like to begin the talk today.
Me gustaría empezar la lectura contemplando estas 4 preguntas. No creo que tengamos el tiempo para responderlas, pero las preguntas nos van a permitir visualizar una cura para los trastornos neurodegenerativos.
Can you do, because it doesn't appear in my screen to say that I'm agree with the recording.
I am recording right now.
Okay, just click on understand, entendido. Otherwise, I don't have it in the, okay.
Good. So we want to ask these 4 questions about envisioning a cure for neurodegenerative diseases, including Alzheimer's. We will proceed with thinking about the answers to these questions. What exactly are we treating? I'm going to talk about that. When do we begin the treatment? When do we start treating disease? Who are we treating? Which patients do we want to treat? And I won't really have much to say about the last question, but it's very important. How do we pay for the treatment? So we will begin here with slide number 1, and that should advance here in a moment, I think. There we go. See.
Let me just see if I can just.
No, it's fine.
Espérate porque voy a hacerlo entendido acá, ¿no? Just give me 1 minute. Okay, yeah, acá. That's it. Okay. Yeah. Okay. See, okay, it'll be at the point of view. Yes. Okay. This one. Okay. Okay, here you are.
Okay, so we will begin by talking about Alzheimer's disease. I believe that many of you will know already a lot of what I will be talking about. I want to give some background here. The purpose of this slide really is that we think about Alzheimer's in 2 different ways. We think about Alzheimer's as clinicians diagnosing the living patient, and we also think about Alzheimer's after the person is deceased from the point of view of the pathology. And this is a difficult match because sometimes our clinical suspicion is wrong. It's not always Alzheimer's disease. And sometimes the pathology that when we think it's Alzheimer's, it turns out to be something else. Not always. The clinicians have become very good at diagnosing this disease, but it's still not perfect. So what we do is as you know, we look in the brain for the amyloid, for the neurofibrillary tangles. And you can see a picture of them here. Neurofibrillary tangles are in the cell body. In this image on top, you can see pyramidal cells that have neurofibrillary tangles.
Inferior. Y en la imagen inferior pueden ver que la proteína tau abnormal empieza a recolectar, particularmente las dendritas que se están muriendo debido a la enfermedad de Alzheimer. Ahora vamos a continuar con esta discusión un poco desde el punto de vista de la patología, o desde el punto de vista del patólogo, que verá Alzheimer con las placas seniles, con justamente las condiciones que mencioné en la enfermedad de Alzheimer. También hay inflamación. Y microgliosis. Pero a veces el patólogo verá mucho más y eso lo vuelve muy complicado, especialmente cuando la persona es bastante mayor. 60.
Have concomitant pathology that goes together with the classical Alzheimer's pathology. And that includes synuclein, which we see also in Parkinson's disease. It includes a protein called TDP43, which we see also in frontotemporal dementia, but we see it sometimes in Alzheimer's. And what gets really complicated is some patients also have vascular disease. That is, they have maybe some small infarcts, they have pathology in their small blood vessels, and that complicates the disease because if we found the perfect treatment for Alzheimer's disease, they would still have their vascular pathology. So sometimes treatments may appear not to work when they might actually work. Now, I want to now begin to talk about another topic. These are biomarkers to diagnose Alzheimer's disease at the earliest possible stages. That's very important because if we want to truly find a treatment for the disease, the treatment has to be started early. If we begin to treat patients once they have advanced disease, many of their brain cells have already died. And that is a condition that's not easy to treat. We cannot resurrect a dead brain cell. But if we catch it early, we do have a chance to be successful. So in this slide, what you see before you are some curves. They are along the x-axis here at the bottom. You can see that's time. That is as the patient is getting worse and worse. And that dotted line is before the person has any, not just no symptoms, but they don't even yet have amyloid deposition in the brain. It's not even Alzheimer's yet. They may be destined to get it. They don't yet have it. And around that time, just when they cross over that line, there are some biomarkers that gradually begin to increase. And the one that may be the most interesting is you see down here in very dark red, it's called plasma p-tau. That stands for phospho-tau. Tau, as you know, is the protein in the neurofibrillary tangle, and it gets phosphorylated at position 181 in the sequence. And when there's a phosphate on that position, then and we have an antibody to that position, that becomes an excellent marker for the disease because there's something about tau when it's phosphorylated at that position. It's an early indicator of the disease, and it's easy to test because you can test it in the plasma. It starts to go up. There are other markers that are better because they go up even higher, but they're more difficult to do because they require cerebrospinal fluid. And they are also various modifications of tau that happen, that modifications due to phosphorylation. Different antibodies detect these forms of tau, and they're very sensitive biomarkers. So we can now begin to diagnose the disease at its earliest stages. Another biomarker is called NFL, that stands for neurofilament light chain. I'm not going to talk too much about it, but it's another good biomarker. It's not very specific for Alzheimer's, but it is a good one. And we can also measure the ratio between two different forms of amyloid, one called 42 and the other called 40. I now want to say a few words about brain imaging, something, again, many people in this audience know about, but it is also a kind of a biomarker. So, as you know, when patients are in transition from the earliest stage of the disease, which we call mild cognitive impairment, then we transition to Alzheimer's. When they have this early form of the disease, we can begin to see amyloid in the brain. And in the slide in front of you, the MCI stands for mild cognitive impairment. And as the mild cognitive impairment gets worse, you can see all that orange and yellow color indicates the presence of amyloid in the brain. Now, look what happens when the patient moves from the high MCI, from mild cognitive impairment, over to actual Alzheimer's disease. Now, the image did not change very much. So while amyloid detection is very good, amyloid imaging is very good for detecting the earliest stages of Alzheimer's, it's not very good for detecting the transition to actual disease. But we have a tool now that can do that, and that's called tau imaging. So look at these images, they're very interesting. And the one over on the left, you see the healthy control. In the middle, there begins to be deposition of tau. That's MCI, or mild cognitive impairment. But notice when the patient transitions to Alzheimer's, now it worsens. So we can actually see that here's a good marker for the transition from MCI to Alzheimer's. And we can do that in the living patient. Okay, now we talked about amyloid and tau, neurofibrillary tangles, in the brain in Alzheimer's. However, there are a lot of other diseases that are caused by tau, which don't necessarily have amyloid at all. And they're listed right here. I'm just going to read them through briefly, just so you have heard the words. And again, many people in the audience know this very well. Frontotemporal dementia is a tauopathy. Chronic traumatic encephalopathy is a tauopathy. Progressive supranuclear palsy is another one. Corticobasal degeneration and Pick's disease. All of these conditions can be diagnosed clinically. Not perfectly accurate, but they're a little different, and it's important that we diagnose them clinically because we have to develop specific treatments for each one of them. So I will just show you here a scan. This person on the right has frontotemporal dementia, and that patient actually, you can see, has a very large amount of atrophy in the frontal lobe. Look how much space is up there where the arrow is. Now let's look over here, and I'm going to show you in this slide some of the most important causes for frontotemporal dementia. Maybe not the most common, but they're very important ones. And those are mutations in the tau gene. You can see in this slide, there are lots of them. And there's many that are listed there. Some of them are in the exons. That means the coding part of the of the gene that codes for the amino acids and makes the protein. And some of them, if you look right around exon 10, some of them are in the region around the exon, which controls the splicing of exon 10. That means whether exon 10 is going to be included in the final protein or not. So we now go, and here, these are post-translation. This is too much information on this slide. I don't like it. I'm going to skip it. It's really a lot of post-translational modifications. Let's go to this slide. So if you take the tau protein that's in the neurofibrillary tangle and look at it under an electron microscope, and you stain it with an antibody to tau, you get an image like this. The little black dots are the antibody, and the white in the background is the tau protein that's in the neurofibrillary tangle. This image is pretty old. We've been able to do this for quite a long time. However, we now, in the last year or so, there's a very advanced technique in which we can now look at these electron micrographs at a much more, with higher resolution, in much more detail. And the insights have been incredible. So here's what happens. So this is now the tau protein in Alzheimer's disease. And what they're showing, this is at the atomic level. What you're seeing here is each one of these dots, those are amino acids, and they show the shape in which they're folding to make in which the tau protein folds to make a neurofibrillary tangle. There's two different ways in which it folds, one on top and the other below. And it is, and those two folds correspond to filaments of tau, whether they're called paired helical filaments or straight filaments. They fold a little differently in Alzheimer's disease. So this level of visualization is incredible to be able to see this atomic resolution. But it gets even more incredible because the next slide will show you now. I hope you can see this. These pictures here in color are the different ways in which tau folds for each of the tauopathies. At the very top, you can see that small letters is AD, Alzheimer's disease. And that's how, and several other related diseases. So tau folds like this, there's a part here, it curves around. But if the patient instead has, see those letters, CTE, chronic traumatic encephalopathy, it folds in a different way. Same protein folds in a different way. Now, look down here below, and you can see additional ways in which tau is folding. For instance, here is a disease I mentioned called corticobasal degeneration. Look how it folds differently. Down here is progressive supranuclear palsy. Each one of these tauopathies, tau folds in a different way. That's amazing because at the atomic level, the way tau folds together is diagnostic for the phenotype. That's pretty amazing because we've come to accept that perhaps we can make a diagnosis of a phenotype sometimes based on a genetic mutation, but never before have we been able to make a diagnosis based on the way a protein misfolds. And that's a very big advance that was done here by these authors, Michel Goder and Shares, just published this year in Nature. One of the biggest advances in the field that we've had. The next slide shows you something we've known for a little while, but it's using now tau imaging, in which how tau spreads through the brain. Tau begins very often in the entorhinal cortex and begins to spread all through the brain, and we call those the Braak stages. It spreads through the brain in a very interesting way because when tau misfolds, it can then move it can then spread to a neighboring cell and cause misfolding in the neighboring cell. That process is called prion disease. And what prions do is they induce the misfolding of tau in neighboring cells. That is, there's a donor cell with abnormal tau. It spreads into a cell with normal tau and causes it to misfold. That's also, there were 2 Nobel Prizes given for that discovery in prion disease, not for tau, but for prion disease. You've heard of these diseases. There's several forms of this disease. And this method of spreading, there's no virus, there's no bacteria. The protein itself is causing the spread of the disease through the brain. And that discovery was a major advance in biomedical science. Now, an important discovery in our lab recently is that what we discovered, also published last year in Nature, was that when tau spreads from cell to cell, the tau, as it moves into the neighboring cell, is taken up by a receptor called LRP1. That was the discovery of our lab. We found the receptor that can takes up tau when it spreads. That's important because if we can find a drug that could inhibit that receptor, at least that receptor for tau uptake, then we might have a treatment. And we're working on that right now. So here is a review of, I gave you a brief idea of some of this. These are the mutations that are found in frontotemporal dementia. By the way, frontotemporal dementia and ALS, amyotrophic lateral sclerosis, are 2 very closely related diseases. Many genes that will cause one, cause the other. Patients often have both conditions. So the mutations are shown here. And we've talked about tau, that's called MAPT, but there are others, progranulin, TARDP, BP, FUS, the expansion of a reading frame in a unknown gene called C9orf. Orf means open reading frame, and a few others that are quite rare that we see down below. Now, here is one of the most common. This is the expansion of a part of the reading frame in this unknown gene. Unknown gene, but when the beginning part of the gene expands with these CG repeats, then it can cause a frontotemporal dementia. We don't really understand this condition very well, but it's one of the most common causes of frontotemporal dementia. So, we have known that expansions can cause disease because we've known for a long time that in the Huntington disease gene, there's also an expansion. So in this case, and in Huntington's disease, it's not a mutation of a single point mutation. It's an expansion where now a part of the genome is grown, it's expanded more widely, and it's made up of these CG repeats. So let's talk for a moment about C9orf. This is maybe the most common form of frontotemporal dementia and ALS. It has, I already mentioned this, an expansion of these GGCCs. All of us have some repeats, but when a person has disease, it expands massively with hundreds of those repeats. There's a concept in genetics called genetic anticipation, which is if the parent has it, the child can get it even earlier because the expansion increases from generation to generation. And these expansions cause very different phenotypes. You can see this down here in the small print. It can cause amnestic symptoms, psychiatric symptoms that are features of frontotemporal dementia. Now, here's another interesting point. I've now categorized these diseases not actually as tauopathies anymore, just as frontotemporal dementia. Because C9orf expansion is not a tauopathy. It actually, the protein that accumulates is called TDP43. Now that's pretty curious. Why would an expansion of this unknown gene cause another protein to accumulate? We don't have an answer for that. I'm going to move on here. Okay, here's another gene that I just finished talking about, TDP43, that deposits as an abnormal protein, just like tau. TDP43 can also cause frontotemporal dementia when you have an expansion of C9orf. But TDP43 can cause frontotemporal dementia and ALS in its own right, even when it on its own. It can have mutations also and cause disease. You don't need to have C9orf. You can have a mutation in TDP43, and those are called point mutations, single nucleotide mutations. So here's the overall layout. This is something I took from a neuropathologist at UCSF named Bill Seeley, in which we can divide these frontotemporal dementias into two types. I talked about the tauopathies. The protein that deposits is called tau. It can fold in different ways and cause progressive supranuclear palsy, corticobasal degeneration. The list is at the bottom. But there's another bad protein, TDP43, that can also have mutations, misfold, and also cause these different types of frontotemporal dementias. And some of them you're very familiar with: behavioral variant FTD, primary progressive aphasias. Another gene mutation here, a gene called FUS. I'm not going to spend much time on this slide, but here, this is now reviewing all of the tauopathies. And I've also thrown in the synucleinopathies. Maybe that's going to complicate things too much. Those are generally can cause Parkinson's disease, in which you get synuclein. So they're a slightly different category, but they're also misfolded, and synuclein also can behave like a prion, in which it spreads from cell to cell. But what I really want to emphasize here are the topography, the anatomy of these different diseases. Here's prion disease affecting the cortex, frontotemporal dementia affecting the frontal and temporal areas, the Lewy body disease, which you're familiar with, Alzheimer's, Huntington's, and ALS. These are the diseases in which there are protein inclusions that cause disease. Of course, there are many other forms of dementia that are not caused this way. Perhaps the most common one is vascular dementia, in which people have many years of, say, hypertension, they get small strokes, and they can get a dementia due to small vessel disease. I want to spend a moment or two talking about chronic traumatic encephalopathy. Now we're again back in the area of tauopathies, because if you have repeated concussions to the head, hit in the head over and over again, then you can get a form of dementia that is caused by those brain injuries, and it is a very severe type of dementia. It has been known for a long time in boxers, called dementia pugilistica, but it's become much more well known now that it's been discovered in people that play American football. And many of the football players in the United States have developed this form of chronic traumatic encephalopathy. In fact, many people that play contact sports get repeated concussions and may be vulnerable to chronic traumatic encephalopathy. The younger you are, when you keep getting hit in the head, the more vulnerable you are to this condition. And it's very serious. People have a progressive psychiatric problems. They become suicidal. This individual American football player committed suicide, and some of them can become violent. I just want to remind you about the difference between concussion and contusion. These people with chronic traumatic encephalopathy may show absolutely nothing on their brain scans because the concussion, when you have repeated concussive injuries, you don't necessarily see anything in the brain. In contrast, a contusion will cause some blood. The picture here is a picture of a person that had a contusion in which they were hit quite hard in the brain and there was bleeding. That's what the white stuff is in the front. But concussions are different. You don't see anything. The person may be unconscious and recover very quickly. They may not even know anything happened. And yet, if it happens over and over again, they can get this disease. I saw a picture of, if any of you are familiar with American football, you have a quarterback who's there who throws the ball. And the people on the line, on the other team, come in and try to knock him down. We say tackle. And this person, there was a movie in which they did it in slow motion. And when he was hit by the other player who tried to tackle him, his head went back very hard and he had a minor concussion. Then after his head went back, it went forward and he had a second concussion. And then he hit the ground with his head and he had a third concussion. In one play, he had 3 concussions. So this can be very serious. And sometimes we can't always tell what happened. Patients who die with this condition have very severe cortical atrophy. You can see that what we call knife edge cortical atrophy. And they have a very typical pattern of tau staining in their brain. You can recognize on the top, these are the sulci and gyri of the brain. And at the depths of the sulci, the very bottom of the sulci, the folds of the brain, the convolutions, the tau begins to collect. That's different than Alzheimer's disease. Okay, do I have a little more time? Good, then I will show you, yes. Let's switch the topic for the last few slides, and I want to talk about some genetics. Here's a way in which you can think about genetics. There are some mutations. Let's talk mostly about Alzheimer's right now because that's what we know the most about. There are some mutations that you see in the top red here that are very rare. They're mutations in presenilin 1 or 2 or amyloid that will definitively cause Alzheimer's. If you have mutations in these genes, you're very likely to get the disease. These are what we call autosomal dominant. They have a very high penetrance. It means if you have the mutation, you get the disease, and they're bad. But fortunately, they're also very rare. So on this x-axis, you see rare mutations that cause very serious disease. Then we have in this middle ground here some variants of genes that will cause that are still quite common in the population. The risk goes down. Look at the y-axis. The risk is now medium, and the frequency goes up. So mutations in ApoE, ApoE4, for instance, are fairly common, and their risk is sort of medium. Then we go into all the way out here, far out on the x-axis, where you see a whole bunch of genes. These are really common variants, and these genes are very, very common. We all have them, but they cause a very low risk. So if you have a lot of these variants in one person, then your risk, of course, goes up. And what we're trying to do now, we as the Alzheimer community, not me personally, are trying to do what's called a polygenic risk score. That is, we count all the variants that a person has in these common genes. These very, very common, but not everybody has them. You may have a couple of the green ones, a couple of the orange ones, but not the others. We add them up and we compute your risk, called polygenic, because it's many genes, polygenic risk score. And this is work that's now going on. Okay, so in the last couple of slides, I just want to, now that we can do this kind of testing, I want to mention how that we have to take this very seriously. There are the reason I say that is because Alzheimer's is still an incurable disease. Some people don't want to know. The information is very serious. You have to be very careful about telling a person whether they have a mutation or not. Some people don't want to know. So there's two types of testing. One is when we already know the diagnosis. That's called diagnostic testing, just to be sure. But predictive testing is what we have to be very careful about. If there's Alzheimer's in your family, you're sure your mother had it. You know from the pathology or from the genetics, and you're the child, you're the son or the daughter, but you don't have any symptoms. We can test if you're in a family with, say, a presenilin mutation. We can test if you're going to get it. Now, we also have to decide whether the gene is a deterministic gene, that is like those presenilin mutations, that if you have that mutation, you're definitely going to get it, or it's just a risk gene. That's another important difference. Another is, how do we assess risk? If I tell you you have a mutation of some sort, but you're not going to get the disease until you're 95 years old, who cares, right? Or maybe some people care, I'm sorry, say 105. So, but if you're going to get the disease when you're 35, that's a very different matter. So not only do we have to tell the person they're a risk, but what is the risk over time? And then if you want to use the information for treatment, you then have to weigh how much risk does the treatment have. Recently there was an anti-amyloid drug approved in the US, but most of us are very reluctant to use it because it has a lot of risk, some risk, can actually induce bleeding in the brain. So all of these are factors we have to think about. Increasingly, we are moving toward what we call precision medicine. This is the answer to the question I posed at the very beginning. Who are we going to treat? All of us are at risk for Alzheimer's, but we don't want to treat all of us. We have to find out who's at the greatest risk. Those are the people we want to treat. So over time, your risk, there's many things that contribute to risk. We know that if a person has a low educational level early in life, their risk goes up. As you get older in middle life, traumatic brain injury we talked about, but hypertension, obesity, all of these also contribute to risk. Later on, smoking, depression, social isolation, even air pollution can contribute to risk. These are all approximate numbers, but if you add them up, we can get a group of people that we think really should be treated. And when you add them all up, you really only account for about 40% of the risk. There's still a lot of information that's missing. Okay, so in this last slide or two, I really just want to end by going back to this idea of a treatment, which is we think is on the horizon. Even though one drug was approved, most of us in the Alzheimer community believe that that was unfortunate because the evidence for it working is not very good. It was very expensive and it has side effects. So most of the people in my field were quite distressed, disappointed at the approval. But in general, as we begin to think about treatments, we think about them in two ways: disease modifying and symptomatic treatment. disease modifying is what we really want. We want to get this disease to slow down, like that blue line, or to at least, or to stop entirely, like the green line. If you only treat the symptoms, those are like the drugs we have now. These drugs are just, they're not very helpful. They will just help a little bit for a short time, see how the patient gets a little better, and then goes right down. So those drugs are not particularly helpful. So in conclusion, I'm going to come back to the first slide. What are we treating? Well, if we're sure it's Alzheimer's disease, then we have one treatment. But suppose the person is very old and they have Alzheimer's disease, but they also have vascular disease, small vessel disease. Maybe the treatment's a little different. When do we start treatment? Pre-symptomatically, or when they first show the very first signs? Who are we treating? Are we treating everybody that's at risk, or do you have to reach a certain risk level before we start treating? And finally, as I mentioned, this drug that was just approved was outrageously expensive. The company set it at a very high price. And this, again, made very many people very disappointed, in fact, quite angry, because we do not want to develop a treatment for Alzheimer's disease that's available only to very few people. Thank you. Do we take questions? Diana, do you want to?
Yes, thank you, Ken. I don't know if you, Carlos, have heard or have seen any questions, or someone wants to question something to Dr. Kosik.
We don't have questions in the chat, but if someone wants to make one, you can hold your hand. I think here in Zoom there is an option, and I can open the microphone.
Yeah.
Okay, if you want, we can go here. Do you have any questions? Like, I have one.
Sure.
When we were talking about treatment, will you say that we could reach the FTD treatment or the tauopathy treatment earlier than Alzheimer's, or it'll be the same race?
I think we don't really know too much about how to treat FTD at this point. Because with Alzheimer's, we can begin to diagnose it early. With FTD, we still have a problem about how to treat it at the very earliest stages.
Just in those cases of progranulin, as we've seen, the probability when we have the mutation already identified.
That's a very good point. Yeah, if we have the mutation, if there's a mutation, like in the progranulin case, we can begin treatment before there are symptoms. Hopefully, the interval between when you start treating and the onset of the symptoms can be a long time. When do you actually, the question remains, suppose the person with a progranulin mutation gets the disease at 45, at 40. If you treat at 40, you have 10 years of treating. It's a long time to be giving intravenous treatment. So we still have that decision, although you're right. With a mutation, we have a little better idea when to start.
Okay, but that will have
It's the same problem, you're right.
Okay.
Same problem.
okay.
So let's see, can you read those questions in the chat?
Okay, yes. Okay.
Buenos días.
Now, buenos dias. No, it's Carlos that it's, if you have any questions, Pablo, or here, the audience, the ones. Sí, sí.
La idea es que pues cada vez hay más manifestaciones preclínicas a nivel comportamental, a nivel motor, que se han asociado pues a la enfermedad de Alzheimer. Digamos que nosotros hasta el momento como grupo no solemos utilizar medicamentos anticolinesteráticos en estos pacientes que no han manifestado pues, la enfermedad. Teniendo en cuenta este panorama, deberíamos empezar a pensar en iniciar medicamentos antes.
No.
¿No?
I would wait for symptoms
Okay.
before giving these anticholinesterase. I would not treat before some. For if a disease modifying drug, anti-amyloid, yes. But for those who are just symptomatic, if there's no symptom, no treat.
See, that is the way the guides, the clinical guides are done, no?
Hay muchos dilemas éticos sobre a qué familiar de nuestros pacientes solicitar o no los estudios genéticos, porque se ven asociados a trastornos de ansiedad, a dudas metódicas, ¿no? Que pueden generar alteraciones en la historia de vida de esa persona.
Sí.
¿Cómo o qué hacen ustedes para decir, bien, a esta persona se lo podemos pedir y a esta tal vez no? ¿Qué ven? O igual se los piden a todos los familiares de sus pacientes.
So this is to ask the rest of you discover someone with a mutation, and now you're asking, do we then solicitar the other members of the family?
Or when to say, when to tell you have something, or because
Yeah.
she says it may produce some anxiety
It's a
and
very good question and very difficult question, because sometimes the family does not want to know. Sometimes they feel it's an intrusion in their privacy. That, now we know there's your cousin or something. So you have to be very careful. But I think what we often do is to try to understand the family tree. And we gradually expand the family tree. So, for instance, there was an older woman who had Alzheimer's, and the son wanted to know if he had it, but the daughter did not. And we were very careful. We did tell her that we were investigating her son, but we told her she does not have to participate. But she does know she's at 50% risk. So your question is very good. I think it's, did you say problemática? It's a problemática.
Yeah.
And I don't have a really perfect answer.
yes. Yeah. Neither do we.
Yeah.
Okay, here is just one question. Is Ana Gama is asking that if it's difficult for people to get the possibility of receiving treatment for Alzheimer's disease, when you think that this type of drugs will be available for the population, the Alzheimer's one, the one that you said that is very expensive,
Yeah.
you said that it should be given a second thought because when to
Right.
give it and this.
Yeah.
But this type of drugs or pharmacological treatment will be able sooner, or even those of the progranulin
Mhm.
mutation, they will have it in the middle sooner or not?
I know it takes a long time. It's right now. I mean, I cannot read the future, so I don't know when we will have these drugs. I think there's good progress. I tell patients that we will have drugs. It's not going to be tomorrow, but the field, the research is moving very well, and that's not perfect news for everybody. They want the drug today, but even the drug that's approved, I would not give it to a patient right now, even if the patient was very rich. I would not recommend that drug. So we have to, we still have some time till we can help our patients with the drug.
Okay, guys, Jose Manuel, do you want to say something or anything? Okay,
Thank
we
you.
want to thank you. This is recorded for the clinical staff that are still assessing patients, because as you know, we have today 5 patients, and if you want to introduce, Sandra is a geriatrician. She's the one who's going to study the program of neuroscience, and she is the one very interested in biological markers and especially on your work. And then if you can go on, she's also.
I'm Valentina. I'm fourth year of geriatric residence and I'm in rotation with Dr.
Geriatric drug.
Yes. These are the two. En
Yeah.
español, pues tranquila.
Soy
Andrea.
coordinadora de enfermedades.
Sí,
Ese coordinator.
sí,
Stephanie Osman, I'm a psychologist.
good.
Okay.
I am Maria Teresa Martinez. I'm also a student from the geriatric residence.
Uh-huh.
In third year.
Third year, yeah.
Gabriel Chingui,
We met.
pediatra,
Yes,
pertenezco al estudio
Red light.
red light, yeah, muy good. Sí, sí,
Functionality.
es un grupo excelente. Ajá.
Yes,
Sí,
but-
we met.
Yes, and then the rest of the clinicians are still in the clinic. So I thank you very much for your assistance, and we will have the recording anyways. Thank you, Ken, for
Thank
your
you.
disposition and the very updated information. Gracias a los traductores, also Diego. Okay, thank you very much. Have a nice day for everybody.
How did it come across in the, I wonder how it came across in the Spanish channel.